In this lecture at Medical Sciences, we're going to discuss about buffer systems and their mechanism of action, or how do they work to maintain pH. So we're going to look at different buffer systems. We're going to define what a buffer solution is. Then we shall give different examples of buffers, and then we shall look at the mechanism of action of buffers. And if it is your first time, if you haven't subscribed, please visit the subscription button below and make sure to subscribe. Then you like the video and share to many others. So about the buffers, the first thing is to know what is a buffer solution. We want to know what is a buffer solution. And the buffer solution, first of all, we have to know the component. A buffer solution should contain either a weak acid and salt of the weak acid, and salt of the weak acid, or it can contain a weak base and salt of a weak base. So this is the composition of a buffer solution. For it to be called a buffer solution, it should contain a weak acid and the salt of the weak acid, or a weak base and the salt of the weak base. And if it is containing these ones, this buffer system should resist change, should be able to resist change in pH when small amounts. The major thing is they should be these components, the weak acid and the salt of the weak acid, or a base and the salt of a weak base, should be able to resist the change in pH when small amounts. The key word here is small amount. When small amount of a strong acid, when small amount of a strong acid or a strong base is added, or a strong base, when a strong acid or a strong base is added. So this is the definition of a buffer solution: containing a weak acid or a salt of a weak acid, or a base and the salt of a weak base, and this solution has ability to resist change in pH when small amounts. If you add much amounts of a strong base or a strong acid, they might not be able to resist. But when you add small amounts, small amounts of a strong acid or a strong base, strong acid which is producing or it is donating the proton, or a strong base which is acting as a proton acceptor by donating hydroxyl ions, should be able. So this is what we can define a buffer: containing these components and is able to resist the change in pH when small amount of a strong base or a strong acid is added. So this is the buffer system. And examples of buffer systems include ethanoic acid. We include ethanoic acid, or you can call it acetic acid, together with sodium ethanoate. This is one of the buffer systems we have, whereby this is the weak acid, this is a conjugate acid, and this is a conjugate base. Then another example we can give is the carbonic acid together with the bicarbonate, and this is known as the bicarbonate buffer. So another type of buffer we can have is phosphate buffer, whereby we have this together with hydrogen phosphate. This is known as phosphate buffer. That is a phosphate buffer. Then another example of the buffer system or solution, we can have protein buffer. And the protein buffer is, we are having amino acid, amine together with ammonium ions. This one is called protein buffer. Then another one we have is the hemoglobin buffer, whereby hemoglobin, we have this stroke this. This one is known as hemoglobin buffer. So these are the examples of buffer systems we can have. And I want to tell you that these last four buffer systems are found in the body. These are known as blood buffers, whereby these buffers, they are the ones that are first line for maintaining body pH between 7.35 to 7.45. So they maintain the body pH between 7.35 to 7.45. And these are the first line. We can call them the first line mechanism of maintaining body pH. First line for maintaining body. So these are physiological buffers. All of these ones are physiological buffers, whereby these physiological buffers, we are going to tell you that among these physiological buffers, we are going to find that these two are found, they are for intracellular, they maintain the pH within cells. Then these two, they maintain in the extracellular fluid. Outside the cell, then this is intracellular fluid. But the phosphate buffer is both. Phosphate buffer can maintain both intracellular fluid and extracellular fluid. So these are the blood buffers. Whereas this ethanoic acid is used in the laboratory to maintain, in the lab, to maintain buffers of solutions or to maintain the pH of solutions, whereas these ones are the body buffers. So after outlining different buffers we have, now this one is going to lead us into what we call mechanisms. So this one is leading us into mechanism of action of buffers. And these buffers, according to their mechanism of action, we are going to have, to begin with buffer number one, which is known as bicarbonate buffer. We are beginning with bicarbonate buffer containing weak carbonic acid and then containing a weak base known as bicarbonate. Or we can call this, can easily call this sodium hydrogen carbonate. whereby this one is going to act as a weak acid. This is a weak acid, and this is a salt of a weak acid. Or this is a conjugate acid. This one is going to act as a conjugate base. Conjugate acid is the one that donates proton, whereas the conjugate base is the one that accepts proton. So if I have a solution containing carbonic acid and sodium hydrogen carbonate, so this solution, if small amount of a strong acid, such as hydrochloric acid is added to this buffer containing weak carbonic acid and sodium hydrogen carbonate, we are going to see the sodium, the conjugate base in this buffer, the conjugate base, that is sodium hydrogen carbonate, being a weak base, is going to dissociate partially to produce sodium ions plus bicarbonate ions. And after producing these bicarbonate ions, remember we are adding a strong acid, hydrochloric acid. So even the hydrochloric acid is going to dissociate completely, since it is a strong acid, to give hydrogen ions plus chloride ions. And these protons which are produced by the strong acid are going to be accepted by this weak base, the bicarbonate, and we are going to end up getting the bicarbonate which was produced is going to react with the hydrogen ion liberated from the acid, and we form weak carbonic acid. So this carbonic acid formed is a weak acid. So we are able to replace a strong acid with a weak carbonic acid. Then after that, we are going to see also that the sodium which was formed here will react with the chloride. The sodium which was formed reacts with the chloride to form sodium chloride. So we can appreciate that due to replacement of a strong acid, which is hydrochloric acid, which was added by the weak acid, carbonic acid, this one is going to help us to keep the pH at a relatively constant pH, So we are seeing strong acid, hydrochloric acid being replaced by, after being replaced by weak carbonic acid, this one with weak acids, they do not affect pH so much. Only strong acids like hydrochloric acid, sulfuric acids affect the pH. So we're going to see, after replacing a strong acid with a weak acid, carbonic acid, the pH of the solution will remain at a relatively constant pH. And this is what we call the mechanism of action, whereby we have got a strong acid which would have caused the change in pH, and we have replaced it with weak carbonic acid after adding these small amounts of hydrochloric acid. So that is the way how this bicarbonate can maintain pH when we add a small amount of strong acid like hydrochloric acid. Then part B of this question, we are still on the bicarbonate buffer. In case when small amount of a strong base is added, e.g. sodium hydroxide, if at this point we add a strong base, small amounts is added to this buffer solution, we are going to see that the sodium which we have added, sodium hydroxide, is going to dissociate completely to give sodium ions plus the hydroxyl ions. And these hydroxyl ions which are liberated, we see even in the buffer system we have there a weak acid, carbonic acid, present in the buffer system. So this weak carbonic acid is also going to dissociate partially to liberate hydrogen ions plus the bicarbonate. And these hydrogen ions which are liberated by the weak carbonic acid comes and combines with the hydroxyl. So the hydrogen ions produced from the dissociation of weak carbonic acid are able to buffer this solution by donating the proton to the hydroxyl ion, forming water. And this water which is formed is neutral. The water which is formed is neutral. So we can see that these hydroxyl ions, if they remain in saturation, they will increase the pH. But we thank God for the presence of weak carbonic acid, which liberate the hydrogen ions, which come and combine with the hydroxyl ions, which would have increased the pH to form, and these strong base hydroxyl ions are replaced with water which is neutral, keeping the pH at a relatively constant level. And then lastly, the sodium which was liberated here combines with the bicarbonate to form a weak. So the sodium ions combine with the bicarbonate. Because remember, bicarbonate is also alkaline, it can easily increase the pH, so it is buffered by forming a weak base, sodium hydrogen carbonate. This one is a weak base. So from this equation or illustration, we can easily see that after adding a strong base, this buffer is kept at a relatively constant rate due to replacement, whereby the strong base which was added, the strong base is replaced by water, which is neutral, hence will not change the pH, and the weak acid and the weak base sodium hydrogen carbonate, which will help us to maintain the pH at a relatively constant level. So in case you are asked in an exam to describe the mechanism of action of any given buffer, you can name that buffer and you tell us what happens if we add a small amount of a strong acid like hydrochloric acid. You write your 4 equations, and also you tell us what happens if we add a small amount of the base. You also give us 4 equations. You can appreciate that each side has 4 equations, and these equations carries much marks. So you have to make sure you have these 4 equations. So this is the mechanism of action of the bicarbonate buffer. So we can end this session by also looking at the mechanism of action of, we can look at the mechanism of action of ethanoic or acetic acid. So this is going to be buffer number 2, which is ethanoate or acetic acid, which is going to act as a conjugate acid together with the conjugate base. So this is the buffer. We are going to look at this last illustration, and we are going to say that if strong, part A is when small amounts of strong acid, e.g. hydrochloric acid, is added to this solution, we are going to tell you that the hydrochloric acid the first equation is hydrochloric acid dissociates completely to liberate hydrogen ions plus chloride ions. And these hydrogen ions liberated, they are the one that can lead to decrease in pH of the solution. So we have to look at a substance in our buffer which can easily remove these hydrogen ions from a strong acid and replace them with a weak acid. That's what we are going to look at. And after liberating those hydrogen ions in the buffer, remember we have the conjugate base, which is the sodium ethanoate, which is going, since it is a weak base, it dissociates partially to give the ethanoate plus the sodium ion. And this ethanoate, which is liberated, is going to act as a base. It is acting as a weak base by accepting the protons. It is going to accept the protons, whereby these protons which can decrease pH are going to be accepted by this weak base to form the weak acid, ethanoic acid. This acid formed is weak. This is a weak acid. So we have replaced a strong acid, hydrochloric acid, with the weak acid, ethanoic acid. And this one will help us to keep the pH at relatively constant level. Then the sodium, which was liberated, reacts with the chloride to form sodium chloride. I told you each side should have 4 equations, 2, 3, and 4, and each equation carries many marks. So that's why we're saying when a small amount of a strong acid, the hydrochloric acid will dissociate completely to give hydrogen ions which have ability to decrease the pH. But these hydrogen ions are buffered by the weak ethanoic acid, ethanoate, which is a conjugate base, which is going to accept the protons which would have increased the pH, and will replace them with a weak acid, which will keep the pH at a relatively constant level. Then another scenario is when small amount, the key point should be small amount of a strong base, of a strong base such as sodium hydroxide, which is commonly used, if the small amount of a strong base is added to this buffer solution containing ethanoic acid and sodium ethanoate, we are going to see that this sodium which is put in this buffer is going to dissociate completely. Since it is a strong base, the sodium dissociates completely to give sodium ions plus the hydroxyl ions. And these hydroxyl ions, they have ability to increase pH to alkaline if left in the solution, but we thank God in the solution we have there a component known as weak ethanoic acid, and this ethanoic acid dissociates partially to give the conjugate base ethanoate plus the hydrogen ions. And these hydrogen ions released here come and combine with these hydroxyl ions to form water. So we are going to see the hydroxyl ions, which would have increased the pH, are buffered by the hydrogen ions from the weak acid, and we form water. We form water, and this water formed is neutral. The water which is formed is neutral, so there will be no change in pH. Then this liberated with the conjugate base combines with the sodium, to form sodium ethanoate. Equation 1, 2, 3, and 4. So this is how ethanoic acid and a sodium ethanoate maintain pH when a strong base such as sodium hydroxide is added, where we are seeing that it will liberate hydroxyl ions which have ability to increase the pH, but because of liberation of hydrogen ions from this weak acid, they come and bind with hydroxyl to form water which is neutral, hence the pH will remain at a constant level. Then also the weak base, the strong base is replaced by the weak base. This is the weak base, and all these two, water which is neutral and the weak base, will be able to keep the pH at a relatively constant level. Hence resisting a drastic change in pH. So you conclude by saying that replacement of strong base, strong sodium hydroxide, by water and sodium ethanoate, which is a weak base, will keep the pH at a constant level, and this is its mechanism of action. So this is what we can discuss under mechanisms of action of buffers, whereby we have looked at the bicarbonate buffer in this video, when we add strong acid and when we add a strong base. Then we have also looked at the ethanoic acid stroke sodium ethanoate buffer, whereby we have seen if we add a strong acid, small amounts, the hydroxyl ions which would have increased the hydrogen ions, which would have decreased the pH, are buffered by the conjugate base to replace it with the weak acid, hence keeping the pH at a relatively constant level. Then if we add a small amount of a strong base, the hydroxyl ions liberated, which would have increased the pH in order to make it alkaline, is buffered by the hydrogen ions produced by the weak acid, forming water. So the hydroxyl react with the hydrogen ions to form water, which is neutral. And also the strong base, sodium hydroxide, is replaced by the weak base, sodium ethanoate, hence keeping the pH at relatively constant level. Thank you so much for learning with us at Medical Sciences. We are always proud of you. Be blessed.